All right. So, yeah, the ending of 15.2, we're talking about how do we control like how do we regulate genes in eukaryotic cells. So we're kind of continuing on in this theme, but we're specifically looking at how we could use something called non-coding RNAs to control gene expression. And I like to think of them as like hecklers. Basically, like non-coding RNAs, they regulate gene. They're just like hecklers. They basically just like talk over the RNA and just like shut them off or like block them. Now, before we get there, it's worth pointing out that of all of the DNA in your cell, like all those nucleotides, A, T, C's, and G's, only 1.5% is actually coding DNA. Now, what I mean by that is actual DNA that would code for mRNA codons. Okay? Meaning, 100 minus 1 is 98.5%. Of the DNA would be non-coding DNA. So most of that is going to be DNA that's coding for some sort of regulation of like gene expression. Some of that DNA will code for these RNAs like ribosomal RNA or the transfer RNA. That's the rRNA and the tRNA that we talked about when we went over protein synthesis. But even more higher percentage of that, I don't know what it is exactly, but a decent amount of it codes for what we call non-coding RNAs. They call them NC, lowercase n, lowercase c RNAs. So let's go over kind of how exactly they would block gene expression. A subset of those we call microRNAs, RNAs, MI. And we're specifically going to look at how exactly they are going to, this is what they do, they either degrade mRNA or they block its translation. They either block or they destroy. And this mechanism for microRNAs that I'm showing you here, this is more or less how the other non-coding RNAs do their job. The details of them can vary a little bit. The protein complex that the non-coding RNAs maybe are a part of can change some. But here's the basic idea. And I think it actually makes some like sense that it would work this way. So let's say again, you have a messenger RNA. You had the best of intentions. You're like, hey, we're going to use this gene and make this protein. But all of a sudden, the body's like, oh my gosh, the environment has changed. We don't need this protein. And you go into emergency mode. It's like, we need to destroy that mRNA before it can make more of that protein that we don't need and could even be like toxic for the cell potentially. So we want to maybe destroy that mRNA. And a way we can destroy it is by using a non-coding RNA called a miRNA, and it'll form a protein complex. So this purple portion right here is the protein. And think of it like when I taught you about the ribosomes. I said the ribosomes was protein and RNA, but the protein in the ribosomes is more of like a scaffold, is more of a structural thing. It wasn't actually really like a functional part of it. And it's a similar thing here. The protein in the mRNA complex is really more of like a scaffold, a structure to hold the actual RNA strand. So the actual kind of action that's happening here is coming from this RNA strand. And so here's how it works. You have an mRNA. mRNA is single-stranded. Well, mRNA, think of complementary base pairing. Let's say this is A, U, C, G. Well, there would be a complementary sequence that can recognize that. That would then be U, A, G, C. So you can have a microRNA that is specifically tailored to a certain gene that could then degrade that mRNA by recognizing, and they say at least 7 complementary base pairings, so I need to extend that sequence at least 7 bases. And then that protein, miRNA protein complex would bind to the mRNA and either destroy it, or it's really the same mechanism for blocking translation. Because think about it, the small subunit of the RNA of the ribosome would not be able to bind to the mRNA if it's already being bound by this miRNA protein complex. So there you go. That's really most of 15.3. Let me make sure I said everything. There is another kind of RNAs that are similar in function to the miRNAs called small interfering RNAs or siRNAs. Again, that last slide, it's really about the same mechanism. It's just technically the precursors they come from, or maybe the protein complex they're part of, is just a little bit different. And this phenomenon, so what I just described here, we call that process RNA interference, or RNAi. So you would say we are regulating gene expression or inhibiting, turning off gene expression via RNAi, via RNA interference. Okay? So make sure you kind of know that vocabulary word and know kind of generally this process here. Okay? That it relies on complementary base pairing. Some other little smaller things. So the siRNAs can play a role in forming the heterochromatin in yeast. Remember, heterochromatin is a condensed chromatin, so it's chromatin that can't be used for gene expression. Now, that happens at the centromeres of chromosomes. Why is that important? Why is the centromeres? Thinking back to the cell cycle and like the phase of mitosis. How would that be related? What were the centromeres used for? Like
during when it was divided, they would find the fibers with
yeah, exactly. Yeah, the centromeres, they held the sister chromatids together. They held the sisters. So during metaphase, the spindle fibers bind, but during anaphase, those spindle fibers bound to the centromeres would pull that apart. In order for the spindle fibers to have something to attach to, you need the DNA to be condensed around that heterochromatin, around that those histones. So, anyways, kind of a smaller detail, but just something that technically exists. And so you have the siRNA system. It not only can be used, and when I say siRNA, the same thing applies for the miRNA. That system can not only be used to destroy mRNA, we can also use it to modify the chromatin with like the methyl groups and the acetyl groups. Okay. And then another kind of non-coding RNA would be pi associated RNAs, pi RNAs. Similar idea, but they're going to be more. They specialize on blocking expression of what we call transposons. This is DNA that can jump. DNA jump. Jumping DNA. It's DNA that can move from one portion of jumping. It can jump from one portion of the DNA to the other. Nor really the AP exam will really test you on that. It's not as important, but basically it's just a kind of another form of non-coding RNA that can prevent DNA from jumping around. The idea was like this maybe was like parasitic DNA that came from like a virus that like can hop around in your genome and cause issues. So there you go. That's the last slide. So then now you have everything you need to finish, I guess, the rest of the mastering bio.